Good afternoon to everyone. I welcome all of you to the 95th lecture in the lecture series in Nonlinear Dynamics conducted by the Department of Nonlinear Dynamics, Bharati Dasan University, with the support from RUSA 2.0. It is my pleasure to introduce today's speaker, Dr. Pushpita Ghosh. Dr. Ghosh got her PhD from Indian Association for the Cultivation of Sciences in the year 2012. She did her postdoctoral studies at Center for Theoretical Biological Physics, Rice University, USA, for about 4 years during the period 2012 to 2016. And she continued her postdoctoral studies also in the University of California, Berkeley, for a year, I think. She started her teaching career as the DST Inspire faculty in TAFR Hyderabad during the period 2016 to 21. She currently working as an assistant professor in School of Chemistry, Indian Institute of Science Education and Research, popularly known as ISA Trivandrum. She has published 24 research papers in peer-reviewed journals and has acquired H index of 11. With this short introduction, now I invite Dr. Ghosh to deliver her lecture. Over to you, madam.
Thank you, Professor Sentivalan, for your kind introduction. And it's my pleasure to be, part of this lecture series. And I have gone through many of these lectures, which is uploaded in YouTube. And because sometimes we cannot, so this is a very good thing that we can go and look at it. And they are really excellent, lectures. And I'm sure students and all our researchers who are interested in this area will be benefited. So let me share the slide. So basically today the topic I have chosen
Uh,
is yes, can you see it, my
yeah, yeah,
slide?
yeah, yes, yes, perfect.
Okay, so the topic I've chosen, it's basically understanding spatiotemporal orders in chemical systems. And to start with, I'll briefly introduce what is the focal theme of my research so far. And over the years, I've been actually working around a common theme of understanding various self-organization phenomena that occurs in different complex systems, either it be a chemical context or in biological context. So what I mean by self-organization is basically evolution of order or formation of ordered structures or functions in the composition of a system under far from equilibrium condition. So this evolution of ordered structures, which can occur both in temporal domain as well as in spatial domain. So example of this temporal orders, which is quite popular and common in mostly in the biological context and physical system, that we could see oscillations, bistability, multistability, chaos, and all of our basic biological processes, be it a circadian rhythm of sleep and wake cycle, or our respiratory, or our glycolytic oscillations, even the heartbeats, those are all where you could see the biochemical changes are happening, and the intermediates concentration changes periodically with respect to the time. So now this is also not this is also very common now in chemical reactions. Those are, there are a set of reactions which are autocatalytic, and those autocatalytic reactions also are able to show oscillations in their intermediates. So there is a famous reaction which is kind of pioneering in introducing this nonlinear chemical dynamics, which is known as Belousov-Jabotinsky reaction. This is a reaction between, bromate salt, organic acid, and a metal ion. Basically, it's a reduction oxidation reaction, which means there are some changes occurring, and this is autocatalytic. And what you could see in this laboratory experiment in this beaker, if you see, there is a change which you can see with respect to this color of this metal ion, ferrous to ferric conversion, and that's appear periodically with time. So this is a very common example of oscillations which you can perform in laboratory settings in a chemical reaction. Now things become more and more rich and very, exotic when this temporal kinetics, which we see where the species or concentration change with time, these are also diffusive in nature, and this kinetics couples to diffusion in space that can lead to various spatial orders in the form of stationary patterns, oscillatory patterns, propagating waves, spirals, targets, etc. The same reaction which you see in this beaker, where we only consider how the changes occur in time, if you try to see it in a petri dish setting, where you could see that how this same reaction, where you could see the chemical concentration is self organizing in a very beautiful manner in like a periodic target like waves, and eventually it will may form in certain condition the spiral waves also. So this is an example of you know changes both in time and as well as in space. So this overall, these things we in a common umbrella we talk about spatiotemporal orders, and my research is mostly oriented understanding these spatiotemporal orders in complex systems. And this is not a simple example. There are many examples where you could see the signature of spatiotemporal orders across nature in different domains. And these are also uh, this spatiotemporal order spans also in length scales, various length scales. So there are some examples I have put on this slide just to show you how from a very tiny bacteria inside a bacteria which is in micrometer length scales, the protein pattern inside the bacteria, you could see the pole to pole oscillations, which is a tiny bacteria. Now think about a collection of bacteria which can form, develop a colony, and as it's very common, then when bacteria colonize it form very spectacular patterns, just like here some of this you could see like periodic structures or some kind of branch like structures. We can go for further higher and higher length scales, to with insects, animals, even spiral galaxies, etc. But here is an example of animal skin pattern which is quite common, everybody has seen in some point this in a zoo or somewhere, like the spot patterns of a leopard skin or a stripes on a tiger or a zebra skins. So these are very common examples in biological domain. The same things we can also see in chemical reactions, those are autocatalytic in nature. Some of these reactions which are performed in millimeter size reactors, chemical reactors, we could see there is a stationary pattern formation in the form of similar to that you see in the leopard skin or a tiger's skin, just like this similar spots and stripes like structure. You could go for further different examples, like a catalytic oxidation of carbon monoxide in platinum surface. Then you could see spiral waves forms, front structures, fronts develops of this carbon monoxide. So these are about some thousand 100 to a few hundreds of nanometer length scales. We can also go some applications where you could see this structure formations self-organized really helpful, while during the formation of interfacial polymerization, these polyamide membranes develop nanoscale Turing structures, just like this blobs and wrinkles, and this actually helps in water purification. So the simple membrane, when it becomes structured, it becomes more and more advantageous over application purpose. So the signature of patterns are not limited here. You could see this in every branch of science. And the common question or quest is that what drives this pattern formation? What are the underlying factors which decides? And if we know those mechanisms, can we, modulate or this modify this outcome in a desired manner? So to address these questions, we need to know what are the driving factors. So without going to any mathematical details right now, we could really see that when I think about a process or is something happening, there would be some kind of kinetics going on, whether some process is forming or changing. So the source kinetics is really important, as well as the dispersal of the components or the species which are involved. And apart from that, there could be some external and internal factors. All this, if we combine, there could be the formation of the spatiotemporal orders in certain conditions. Now, being a theoretician, this is more advantageous to understand and predict by theoretical formulations how you can actually understand the mechanism of pattern formation by write down you developing some models. And in most of the cases, the frameworks what we utilize are of continuum based modeling or of particle based modeling. In the continuum based modeling, what we can denote the concentration or the density of this species which are involved in the process in terms of certain you know chemical certain time evolution equations, which where we can utilize the tools of nonlinear dynamics to understand the stabilities and to get to know whether the system can actually show certain spatial orders or not. And we definitely utilize the numerical techniques. There is another sort of models which based on specific things and depends upon the length scale or the type of the problem that you are interested on, where we could also look for the individual's interaction, their interaction in the terms of forces. And one can actually utilize the molecular dynamics same as the dynamics, which means we try to understand the evolution of the particular species, and there we use mostly these evolution equations and use a statistical mechanics tool. But today I'm mostly focusing on the continuum based modeling, just like this Petrus's experiment or the Becker's experiment, where you could see a chemical reaction is happening and whether it can lead to the spatiotemporal orders. So briefly again, these continuum models will have a reaction part, a diffusion part, or it may also have some drift part because of some external factors. We write down the equations of the species, their time evolution, in terms of the partial differential equation, which also takes care of their spatial extension, meaning the spatio-temporal evolution. And then we utilize the nonlinear dynamics and numerical techniques to understand the evolution process. So that's the whole framework. And as talking about, you know, pattern formation, the very first idea and which is like a most popular and fundamental was proposed by Alan Turing in about 1952. So he provided a chemical basis of morphogenesis, mostly in the biological context, but this mechanism is widely used in almost every branch of science and it's very, very fundamental. So the whole idea is when there is a reaction or some sort of source kinetics is there and it couples with the dispersal processes like the diffusion, it may lead to spatial pattern formation through symmetry breaking. And what does it mean? Our intuition, general intuition says that if there is, some inhomogeneity in space, for example, if you open, a scent in a room, with time it gradually tried to equalize the space and homogenize. So basically diffusion alone try to homogenize the system, making a uniform concentration throughout. But when it interacts with reaction, there is a possibility under certain conditions that it can form sustained inhomogeneity and develop spatial patterns in the concentrations. So this, in more formal words, in terms of nonlinear dynamics, it tells that a homogeneous stable steady state under diffusion becomes unstable, and a diffusion driven instability gets to form stationary patterns. But it is it depends upon certain conditions. So the conditions are like that in a interacting two variable system. Let's say there are two interacting species, one, and they follow activator inhibitor kinetics. So the reaction is, let's say, autocatalytic, and one of the species activates its own production or inhibits, activates inhibitor also, and the inhibitor inhibits the activator. And at the same time, if the inhibitor diffuses faster compared to the activator, at such certain condition, it may lead to the diffusion driven instability. So this is the whole idea, and it seems apparently, very simple. But achieving these conditions in realistic situation in a chemical reaction is not very easy. So just to give a brief, theoretical background of Turing pattern formations. So in terms of reaction diffusion equations, let's say U and V are the two components, and F of UV and G of UV are the reaction kinetics. That can be some sort of nonlinear, cubic, or some more complex reactions. And these are the diffusion parts. So du and dv are the diffusion coefficient of the activator U, and let's say the V, or V species V, dv is the diffusion coefficient. And now, just utilizing the nonlinear dynamics tool step by step, we can find out the homogeneous steady state of the system. And then our goal is to study whether this homogeneous steady state, which is otherwise stable because of the temporal perturbation, may it lead to can it break the symmetry in, when it is parted by around the steady state under a small, infinitely small spatiotemporal perturbation. So basically we can write down this kind of situation where the perturbation delta u and delta v corresponds to the perturbation around the steady states us and vs. And this perturbation can, we can take a form like e to the power lambda t and cosine parts, so which has a temporal growth parameter as well as spatial extensions, and this only depends upon the reaction parameters. Now utilizing this, you can simply linearize the system and find out the Jacobian matrix around the steady state, at the steady state. And then we could see the stability conditions, whether the trace of this Jacobian matrix is negative, and this situation the determinant of the matrix is greater than 0. These are the conditions which make sure your homogeneous stable steady state is stable. Now this actually leads to the situation which we described in which Turing proposed, that the system needs to be autocatalytic nature. So one can act as activator, another can act as inhibitor according to this kinetics, and there should be a large disparity in the diffusion coefficient between these two species. And particularly here you see that diffusion of the inhibitor V has to be much larger than the activator. Under such condition, you can see the dispersion relations, and you could see the real this eigenvalues or the growth rate of the perturbation can become positive, and it can result to the stationary pattern. So this is the briefly the theory behind the Turing pattern. Now as I mentioned, that achieving this condition in realistic chemical system is not such easy. It can actually, because most of the cases, this disparity of the diffusion is the problem. Most of the cases, they are very, close. So over the years, theoretical and experimental efforts have been taken to, to utilize some external perturbations in forms like in different ways, so that it can actually overcome this stringent condition of Turing instability and may lead to the Turing like pattern or diffusion driven instability. And chemical system in that case are actually beneficial, and they have advantage over many biological system because we can manipulate, and they can really susceptible towards many external perturbation. And so it basically took almost about 40 years after the discovery of this Turing's, condition and Turing's pattern, theory of this reaction diffusion Turing patterns, that it took about 40 years to realistically come up with actual experiment where you could see this to evidence of Turing patterns. And this reaction, this is a reaction which is known as chlorine dioxide iodine which is developed by Lengin and Epstein group in 1990. um, so the whole setup is, just to give you a brief idea of how the experiments are being done. you we take a very thin almost two-dimensional thin slab which is embedded in acrylamide amide gel, and these two faces of this gel reactor is attached with continuous star tank reactors which has the the initial reagents, the reagents like malonic acid or chlorine dioxide or the iodine. So when they come in the contact, and we in this thin slab, the convection is not possible, it's we can eliminate, only two-dimensional diffusion can be considered. So this is kind of a setup is used in an experiment, and the reactions are actually complicated. There's a many step autocatalytic reaction because these are the initial reagents, malonic acid, chlorine dioxide, iodine, and these undergo several steps of multi steps reaction. But these are the key process that I have shown here, that this malonic acid reacts with iodine and it forms iodide, and this iodide then again reacts with the chlorine dioxide, forms a chloride, and the other steps of the reactions also happen. So basically it's an autocatalytic reaction, and it it has been noticed that main players of these reactions, this iodine iodide, chloride, these are the intermediates, and that decides the overall fate of this reaction. So the experimentally, when they this group castrates and co-workers, they basically realized this experimental Turing patterns in this reactor, in this type of reactors, where you could see the spot like structures have come up, which are basically the bright spots are the iodide, which is one of the intermediates which acts here as an activator. Now this um, this group, Lengin and Epstein, they come up with a phenomenological description of this reaction, this experiment, and they come up with a model, which is definitely a reduction, but it's it's basically supports very well all the experimental evidence or observation that we see in this particular reaction. So this iodide and chloride are the key players. Those are the intermediates of the reaction, and this we denote as U and V. And these are the basically the reaction kinetics. So this is a kinetics which you could see where A and B are the parameters which are comes from the malonic acid, chlorine dioxide, or iodine, which is a large excess. Those are the reactants, initial reactants. So, and the sigma that I have to mention later, which has a very big role to create the disparity in the diffusion coefficient between this iodide and chloride ion. When this starch is put in this reactor, this iodide ions form a triiodide, which reduces its speed, now the diffusion coefficient, and that's how they make out this condition of disparity in these two species. And this entire system can be written in this sort of reaction diffusion model, and if you numerically simulate it, you can realize a similar kind of mimicking Turing patterns. So this just to give you an idea about this reaction, which is very pragmatic and versatile, and it's a lots of application of this reaction to realize different types of spatiotemporal orders. And I have used this reaction, this model system in various of my works. And as I mentioned, that's getting now coming back to the again this chemical system that I mentioned, that how you can achieve such a disparity and diffusion coefficient on this to overcome the conditions of this Turing instability. People use, researchers use different external or internal perturbation in the form of time delays, electric field, photo illumination, fluctuations, different kind of directional flows, temperature gradients. In some sort of some species, there are chemotactic gradients also been created, and these all can contribute in spatiotemporal orders in various aspects. So today I mostly will talking about these two such external perturbation, electric field and temperature gradient, which can be effectively utilized to develop different a diverse variety of spatiotemporal orders. Before that, I just briefly mention that being photosensitive in nature, this particular reaction, this chlorine dioxide iodine malonic acid system, it is susceptible to photo illumination, and it undergoes a reaction where you could see we can utilize this kinetics and the dynamics, where you could change this illumination parameter, and you could realize the Turing patterns, the spot-like Turing patterns undergo transition to a labyrinthine stripe-like patterns to again some inverted spot as you change the increase the illumination parameter, illumination intensity, and for a very high illumination it becomes homogeneous. And in my doctoral work, I have also worked on this numerically and using the nonlinear dynamics tools. I carried out some nonlinear analysis using Galerkin skins to realize this side of different types of patterns. I have also worked on another very interesting part in spiral wave formation and its effect under periodic illumination. So what we can see, a temporal forcing through periodic illumination can create a spatial changes by dense and sparse kind of spiral wave formulation with respect to temporal forcing by photoillumination. And it has been seen that this phenomena of with time mimics with his the frequency of this change of dense to sparse spiraling happens in the same frequency when the frequency is similar to the periodic illumination of this light. And we carried out multiple time scale analysis for to realize all this. And I'm not going to the details, just to give you how versatile this reaction is and how we can utilize the a simple external perturbation to realize such diverse spectacular dynamics in a chemical reaction. We can also utilize time delayed feedback in pattern formation, which through this photo illumination, where you can see that a suitable time delay and the strength of the feedback can realize this kind of stationary Turing like patterns. So now let's move on to today's topic, where I wanted to tell you about how we utilize electric field and temperature gradient to realize the spatiotemporal dynamics of chemical reaction diffusion systems. So thermodiffusion, so this is some phenomena that many of you have heard as a Sore effect, where mass transport is induced by temperature gradient. So let us just look at this schematic, where you have, let's say, some species, some in a reaction chamber, which is a kind of a square in rectangular in shape, and you maintain one side of this reactor with a hot reservoir, like temperature is high, and the other side you maintain a cold reservoir to create a certain constant temperature gradient across this reaction mixture. Now the question is, so this temperature gradient actually has been utilized, this thermodiffusion, in many applications to segregate different components. Like some of the species move towards the hot end, or some of the species may move towards the cold end of the this reaction chamber, and you can have the segregation. This there are many application so far in past people have used thermohaline convection, purification of isotopes, separating metals from alloy, and also different kind of wave instabilities. So the whole idea is whether a reaction, a system which undergoing this reaction under this temperature, constant temperature gradient, can we can create or modify spatial patterns or not. So this is the whole question that we wanted to address in a very systematic and general way. So if we think about it in a reaction diffusion framework in a two-dimensional setup, this is the time evolution, space time evolution equation, where you have u and v, two species, let's say, which follows a reaction kinetics of f and g, and there is a flux of this species j and ju and jv. Now while maintaining a certain temperature gradient in this situation, we considered this temperature effect doesn't form create any reaction or changes its kinetics. It's only affects the dispersal of the species. So mostly the flux is being um, modified. So if you see as I species flux of the ith species, which is having a part which is normal diffusion and a part which comes through this temperature gradient. So because of the mass transport, because of this temperature gradient, so this basically modifies the flux. And you could see there are several parameters. You could see dt, d is the normal diffusion coefficient, dti is the thermal diffusion coefficient, and ci is the concentration of I species, ri is the relative concentration of the I species, ci over sum over ci. So let's say we have this two species system, then it will be c of u by u plus v, just like that. dt is the temperature diffusion thermal diffusion coefficient, which is proportional to the normal diffusion coefficient by a coefficient which is largely known as Sore coefficient or Ludwig-Sore coefficient. This st of this ith species can also depend upon the concentration of the species, and it has a form like this, where this case is a phenomenological constant, and there are literatures from where we could actually get these formulations, and we utilize this effect. So basically altogether, if we took if you consider this modified flux in this reaction diffusion setup, we could see this kind of spatially extended system under the presence of thermal gradient, when stu and stv are just the Sore coefficient of these two species u and v. Chi is nothing but the sum over of these two species, which is the initial constant, which we can just have a free parameter we can take as one for our purpose. Ks is a phenomenological constant, and we could have this values of this constants from the literature. So we have chosen those parameter, and this delta t term, or this gradient of t that you see here, this is actually a constant. It is not it's just appear as a constant term. So across the reaction mixture, across the time evolution, we just kept is a constant. So now we follow the usual procedure. Find out the steady state of the system, homogeneous steady state without any temperature gradient or without diffusion, carried out a linear stability analysis with and without diffusion, find out the stability matrix, eigenvalues, and find out whether there are real and imaginary eigenvalues, meaning the growth of the perturbation. And from there we can if the systems are not very complicated, we can find out the Hop and Turing bifurcation curves, and from there we can see how this temperature gradient actually modulate the eigenvalues and lead to certain situation where the homogeneous steady state which is otherwise stable in presence of normal diffusion becomes unstable in presence of certain temperature gradient. So that's the whole idea, and we basically applied this in two model systems. One is known as the Bruslater model, and one of this I already mentioned, chlorine dioxide iodine malonic acid system. So let's briefly talk about this Bruslater system, which is a theoretical model, very early theoretical model of autocatalytic reaction proposed by Prigogine and co-workers. This is the reaction schemes is kind of follow these steps where you have reactants like A and B, and you could from there you can get the two intermediates, which is X and Y. This reaction is autocatalytic. You can just write down the equations from here, which comes like this: the changes of X and Y with respect to time in terms of our my convenient variable U and V. I just write down this kinetics once again, where A, B are just parameters which depends upon the which are present in a large excess. So these are just like the parameters. So you can find out the Hopf bifurcation and Turing bifurcation curves following the nonlinear dynamics tools here, carrying out the linear stability analysis. And this picture here, this photo, where we have plot the B versus diffusion of the activator. These are the Hopf line. The blue line is the Hopf line, and the red line corresponds to the Turing line. And as we see from the linear stability analysis, the region below the blue Hopf line and above this red Turing line corresponds to the region of diffusion driven instability, where we can realize the Turing patterns in the form of spots and stripes. And now this is a point which O2 O3 I have chosen here. These are the points where these are Turing region, and there is a point mentioned here in this bifurcation diagram by O1, where you could see the value of du is 5, which corresponds to the situation where diffusion driven instability is not possible, because the diffusion coefficient of both the activator and the inhibitor, which here you could see the dv is also 5. So the situation, so we are mostly interested in this points where you have the no diffusion driven instability, but with respect to this presence of temperature gradient, whether this parameter, this this state becomes unstable and gives rise to any Turing instability. So with that, we tried out with these equal diffusion coefficients. And now the question is, when we have an equal diffusion coefficient, and I didn't mention again that there is this parameter Sore coefficients STU and STV, the sign and the magnitude of this Sore coefficient actually tells you about the mobility around this hot end of the reaction chamber or towards the cooler region of the reaction chamber. So the positive signs basically tells you that the species moves toward the cooler region, and negative signs implies that they are moving because of the thermodiffusion, they're moving towards the hot end of the reaction chambers. Now if I keep both this coefficient same, and most of the cases there is not much difference in the Sore coefficient of the ionic species. So in that situation we are not able to create any differential mobility, and it cannot give you any inhomogeneity. So in another way, we can realize this differential in mobility with respect to the difference in the Sore coefficient. So that's what we have done here. You could see when there is no temperature gradient, the real eigenvalue, this is a 3D dispersion curve along kx and ky. This is negative, and as we incorporate this this system becomes under the temperature gradient, it realizes real positive eigenvalues, and you could see the numerical simulation that also corroborates this idea from the linear analysis of this point. When there is no temperature gradient, that is delta gradient of T is 0, it's a homogeneous pattern. When we have the temperature gradient, it realizes stationary pattern, Turing like stationary pattern. And as you can also see from this figure E and F, that as I keep upon increasing the temperature gradient, this constant temperature gradient, you could also not only see the formation of pattern, but also you could see there is a definite changes in the wavelength of the patterns. So we also see the similar situation when we could take, because we don't know a priori, there was not any experiment performed in this system that whether this can be positive or negative. So we have all the we you carried out all the possible combinations, and if we take another possibility where you have both the signs negative, then also we realize the similar stationary patterns. Now, you coming back another here, that whether there is a possibility that one of the species is moving cooler region, another species is moving in the you know um, uh, this hotter region, then we have to take a opposite sign of the this Sore coefficient, and then we find that it at a higher value of temperature gradient, it also lead to the stationary patterns. I am not going to show all the details of the all the results, just to mention. So now we come back to another points. Those are like O2 or O3, which are basically already in pattern state. So here we realize the Turing pattern. So we wanted to see how the pattern transition happens in presence of certain, this type of temperature or thermal gradient. So in this situation, we chosen same coefficient of Sore coefficient, because this is already in a diffusion driven region. So there is already a diffusion disparity between these two species. So we took same value of the coefficient, and we realized that whether this is stripe like or spot like Turing pattern, the finally with the presence of this temperature gradient, this undergoes a transition like vertical stripes, and then it for further value of delta T, it basically form this bent like structure, but both are stationary patterns. So this is something very interesting, that it tells that we not only can create spatial patterns, as well as we can modulate just simple presence of a temperature gradient can give you diverse types of patterns as you desire. If you want a vertical type of pattern, you can have certain value of that, and you can modulate these. So basically, it's just coming to the next system, CDIMA model, the same thing. I'm not going to describe. There is a point which is above the Turing region, which is a homogeneous, where you cannot realize the pattern. The other one is inside the Turing region, where you can see the Turing patterns, and we wanted to see what happens in these both points. So when we take same sign, either positive or negative, same sign, the same magnitude of the values, we find that from the homogeneous this point, we can see formation of or development of stationary patterns, and with higher values, it find of end up with this bent like structure. In the Turing region also, we could see these spot like patterns undergoes transition, and also finally end up with higher value, this kind of bent stripe like structures. So basically, generation and transformation both can be seen when we you have taken a same sign of the Fourier coefficient of both activator and inhibitor. And then we wanted to see what happens if we, take an opposite sign. So we chose one of the species activator moving, cool end, and the other one, hotter end. And then what we see with this point, which is otherwise homogeneous and stable, shows uniform pattern. It shows you some sort of moving waves. So can you see these waves? Yeah, so basically, when we take opposite signs of this Fourier coefficient, that says that one is moving in opposite direction with respect to other because of the thermal diffusion, we could realize propagating and oscillating spatial patterns. So you could see this moving, spot-like structures moving in certain direction, and this kind of patterns also, moving. So these are like very complicated moving patterns, and we can realize this with respect to comparing the real and imaginary eigenvalues, whether which one is dominating over what. So when the imaginary eigenvalues dominate, we could see this propagating or a wave like instabilities appearing. So now to mention that so far I have already shown that we have chosen a region where there is a disparity in the diffusion. But what happens, and this thermodiffusion solely cannot create, the instability in the form of stationary or non-station wave instabilities. Because of this condition, if I choose this diffusion coefficient, the ratio of the diffusion coefficient d, both the activator and inhibitor same one, and there is no presence of stars, the Hopf and Turing lines are separated to a very large region, and there is no way we can realize or lift down this Hopf line below this Turing line. So it's not possible without this either the presence of starch or this disparity in diffusion coefficient. But in this situation, when we realize this simulation, we see there is a spiral pattern formation. And then under this temperature gradient, what we see, this spiral wave show a drift motion from one end to another. You could see these waves are moving in presence of the temperature gradient, which is non-zero. And we could see this spiral patterns, when it get hits to the boundary, it basically diminish. So basically we could see a drift motion of the spiral waves. But when we choose the opposite sign, we don't see a drift motion, we realize the stable spiral patterns. So this is all about the variety of patterns that you can simply find out because of presence of a temperature gradient. And just to summarize the same things, this thermal gradient can be effectively utilized and create and modify the concentration patterns. You can see stationary patterns, you can find oscillatory patterns, we can see drifting motion of the CDMA system. So for further details, I refer this publication. You can get all the details of all these pattern structures and everything here. So let's quickly move on to the next topic, where I wanted to show you how mobility can be also utilized in forming the diverse spatiotemporal instability in autocatalytic chemical reaction. And as I mentioned, these chemical reactions, most of the cases, these are ionic in nature species, like this chloride iodide I show. These species are susceptible to external applied electric field, meaning ionic species can move across or along or against the applied electric field. So in presence of the electric field, mostly the flux of the species get modulated. And in addition to the normal diffusion, it has a part which also comes through because of the presence of this externally applied electric field across this two-dimensional setup, as you just seen like the temperature gradient we created. So this is a form of the mobile flux because of the mobility, ionic mobility, and these are the expression. I'm not going to details of this. So basically, if you work down this entire thing, this flux, it can this Ex and Ey are the electric field component along the X and Y direction, and it can incorporate in this two-dimensional reaction diffusion framework with the normal diffusion part and a part you know concentration gradient because of the electric field. So here Zu and Zv are the charges of the ionic species. Ex, Ey, as I mentioned, the components along the x and y direction. And when we just make it simplified, Ex and Ey, these are the strength of the electric field in the x and y direction. So one, this is the complete spatially extended system in presence of this bidirectional bidirectional electric field, presence of this electric field, and now the sign of these charges and the strength Ex and Ey basically will decide the movement of the species along against the constant gradient imposed by the electric field. And our aim is to find out the conditions and realize the patterns, spatial patterns in presence of the this electric field. So we chosen the photosensitive version of CDIMA system, just this phi is illumination parameter, and these are the same diagram I also shown earlier with A, B are the parameters, sigma the starch, phi is the photo illumination, and these are the regions where you could see homogeneous, or these C2, C3 are the points which are in the Turing region. So now in the presence of electric field, we can derive the conditions, the 3D dispersions, and you could see when there is no electric field across this two direction, we have seen the eigenvalue comes as negative. But as soon as we have the this presence of electric field in the system, we could see the presence of real positive eigenvalues. And one can also think about the presence of unidirectional field and bidirectional field. And this is the point I have chosen, C1 point, which is homogeneous, stable, that you could see no pattern when there is no electric field. But when we have this bidirectional electric field, sorry, the unidirectional electric field across the x direction, where the component Ey are 0, you could see there is a development of stationary patterns, and finally it takes up certain values. As well as you increase the value of this intensity or the strength, the wavelength also changes. Now the same situation, point, when we numerically simulated under bidirectional electric field, it undergoes the pattern formation in a different way, and you could see a bent like structures coming. But another interesting thing we have seen, that whenever there is a one of this strength of the electric field is higher in certain direction, the patterns basically align or appears along that direction. So when we have a higher value of Ey, the vertical stripes will come. So the same situation can be seen if we just change the Ex and Ey in the opposite manner. So this is pretty similar, just you will see the opposite directions. No, now the thing is that there is this Ex and Ey, the strength we wanted to see how, and because for this given charges of iodide and chloride, we know those are negative, we cannot change anything there. We only can change what happens with this modulation of strengths, and we could see that average deviation of the the one of the variables activator or any of this, we can find out the deviations or the variance of these values around the steady state. And you could see that basically this system is largely influenced by the presence of electric fields, and you could see a wide parameter range where you could realize this kind of patterns. So now we also look for the pre-existing Turing pattern, how it deforms under electric field. And as you see, these C2 points have the spot-like structures. It undergoes electric bidirectional electric field. And when I change one of the field strength is high, it comes to show you the vertical along that direction. Here we have chosen y direction, and you could see as I increase the electric field intensity, ex and ey, here you could see there are clear change of the wavelength. Similar phenomena you could see from the point C3, which undergoes from the labyrinthine Turing pattern to this kind of similar vertical stripes. So basically here also we could see the changes. Now let's see another model, because kinetics is also very important in this CDIMA system, because this is a realistic chemical reaction. We know the species charges of the species, they are negative, and we basically investigate how the strengths of the electric field intensity modulate and develop and modulate the pattern formation and transitions. Now this Brusletter model I earlier introduced you. Here also we come up with linear stability analysis, and we could see a Hopf bifurcation curve and Turing bifurcation curve. And this region below the Hopf curve and the Turing curve is the Turing region, where you could see the stationary Turing patterns. And here the point B1 and B2, where you could see where the diffusion coefficient of the activator has been taken again same as the inhibitor. That's at the point where we don't see any diffusion driven Turing instability, and our initial focus is to see realized patterns under electric field in these points. So we have seen that when we choose same sign of the charges, because we don't, this is a theoretical model, we don't see any symmetry breaking, because the same sign of the charges cannot create differential mobility because of the electric field. Then we chose a different sign of the charges, and we could see that with that this easily breaks the symmetry, and in the presence of this electric field strengths in both the direction, you could see the real we could see real eigenvalue as well as imaginary eigenvalue. So there is a difference between compared to the CDIMA model. There you could also see a presence of imaginary eigenvalues, and the competition between this real eigenvalue and the imaginary eigenvalue will actually decide the fate of the final pattern, whether it will be a stationary or moving. So these are just the real plots of the real and imaginary eigenvalue with respect to the wave vectors. So now chosen this B1 point where we have equal diffusion coefficient, and the condition of differential mobility created through taking the opposite signs of the charges, we could see development of wave like instability, and that's further confirmed from the the dispersion relations, that the imaginary eigenvalue actually dominates over the real eigenvalues. I didn't show you this movies here, just to tell you these are basically moving patterns, these vertical moving patterns, as we keep on increasing this unidirectional electric field, where there is no bidirectional, only the x direction, along x direction we incorporate the electric field. With a higher value, we could see there is definitely coming up a large homogeneous region, and we realize that around a value around 3, this entire system 3 greater than 3, this becomes again homogeneous. So there is a definitely a modulation because of the presence of the electric field, such that we from a homogeneous state we realize a wave instability. The same situation, traveling wave like pattern forms when we have a bidirectional electric field. So you could see here the values. When we have the same values along the both direction, we could see these bent like structures, but one of them, when it is higher, we see this vertical type of structures. So you can easily manipulate how, the type and the waves, whether you want a vertical stripes or a horizontal stripe like inhomogeneity moving. So just tuning these directions of the electric field strengths. So now when this so now I talked about the same opposite signs of the charges, but there is another way for by which we can also create the differential mobility by taking the same sign, but let's say we have the different charges. So that can also provide a way to having a differential mobility. So in this context, we chosen a charge, let's say -1 and -2. Most of the ionic species are generally -1, but there are species, some ionic species which have -2 charges. So with this situation, same with the diffusion coefficient and the same this um, well, there are these points. I will one more time show you these points b1 and b2. We can see the initially when we have a small value for the, this is for the b1 point, we could see a traveling wave, and then as I increase upon this electric field intensity, we could see that there is a appears a stationary pattern. The same thing is with bidirectional electric field also. With low values we could see traveling waves, but as long as we have a higher value of electric field intensity, we could realize stationary patterns. So this is something definitely can be realized only comparing the imaginary and real parts of the eigen values. And it's really hard to just simply tell you about through linear analysis what and kinds of patterns will appear. So for that we should do a nonlinear analysis, but which is beyond the scope of this particular study right now. So we only compare this eigenvalues, and that very well corroborates with our results, numerical simulated results. So now and we're coming back to how we realize the as we change this ex and ey, the strengths, where we can see the in which parameter sets we could realize the pattern. So the same thing, we plot the 2D heat map of this average deviation from the steady state, and we could see there is a this a is for the point b1, which was kind of away from the bifurcation line here b1. We could see there is a very small region of both the ex and ey where you could see the patterns, but when we increase the strength of this electric field, we don't see any pattern. So it is very clear that we have a limited region where you could realize the patterns, and that's also corroborated with the numerical analysis where we don't realize the patterns. Now coming back to the other point b2, which is a little bit closer to the bifurcation line, we could see this gives you a with this same sign but different magnitude, we could see there are the pattern formation with respect to ex and ey, and there is a very kind of bigger domain where you could have this ex and ey strengths varying, and you could realize the patterns even when the diffusion coefficient is same. So what we see here, we see here stationary patterns, and here we basically with the opposite signs we only realize the uh, traveling wave or moving pattern. This is not able to show you any stationary pattern, this b1 point, but b2 point can see with the same sign even the stationary patterns. So basically now coming a little bit more digging upon the other points which are already in a Turing region. So we also wanted to see how versatile this effect of electric field intensity is. So from the spot-like structures, what we realize, it basically develops very interesting moving patterns, type-like moving patterns. One of them are shown here as a movie. You could see it's coming up, it's basically breaking, and then again it's moving. So it's a very interesting kind of very complicated patterns are coming, and those are basically all either unidirectional electric field, or you can choose a bidirectional electric field, and you can realize different kinds of patterns, different kinds of wave instabilities. Similarly, talking about the other point, which starts from a labyrinth type, it also undergoes pattern transition from stationary pattern to wave instability in the form of stripes like moving pattern. And you could see something like this, very nice, which is kind of aggregated structures, and then it moves. I don't have the movie right now here, but this basically moves. So these nice moving patterns you could realize because of just simple interplay of the intensities along x and y directions. So, to just summarize briefly, we realize that the presence of this electric field in externally applied electric field actually can lead to develop pattern formation from a homogeneous state, and it can also give a wide variety of diverse moving patterns, some of which I discussed and showed you in this top my today's lecture. And one of the important things is that the sign and the magnitude of the ionic charges actually regulate this differential mobility and decides the fate of this outcome of the pattern state. And this work is just last year in December has been published in this journal, and you can have the details of all the entire thing. And I acknowledge my first PhD student Arpan, who has worked on this project. And we are further looking at to because of this chemical system, how different kinds of electric field like circularly polarized electric field can have an effect, the dual play of illumination and electric field, so which can also give a very nice variety of diverse spatiotemporal orders. And as long as we can realize, we can modulate, we can modify as our desired outcome. So at the end, now I will, stop and acknowledge. So this entire area of nonlinear dynamics was introduced by Professor Devshankar Rai when I was, working under her supervision during my doctoral work, and that's what I got interested, and still I'm carrying out these works, and I'm sure that this is known not only the chemical reaction, but there is lot of potential to work on this biological system, where this spatiotemporal orders are very very common, the active systems. And I also acknowledge my postdoctoral advisors, who also, where I also have worked on biological system and under investigated spatiotemporal orders in biofilms, bacteria. So finally, these are the funding agencies which has supported me throughout the years, and I thank you all for your patience and kind attention. I took a little bit larger time to stop, so I will be very happy if you have any question. Thank you all.
Thank you, uh, Professor Ghosh, for your very interesting talk. So now the forum is open for questions, clarifications.
Can I ask a question?
Yes,
Sure, sure.
Professor.
Okay, maybe a couple of short questions, general questions. One is, in the reaction diffusion system, is it possible to have a stable two-dimensional pattern that would be not periodic but quasi-periodic? For example, something like Penrose tiling or something else which is spatially quasi-periodic. And another question concerning spiral patterns, rotating spiral patterns. Can it be stable if the topological charge of the spiral is larger than one. For example, if you have a double spiral.
Actually, I mean, various situation can occur, and as I mentioned, the whole of these things is depends upon the reaction kinetics as well as the diffusions, right? On the presence of this external perturbations. So, for this, for example, this CDIMA system, chlorine dioxide iodine malonic acid system, when we realize a stable spiral pattern where there is no diffusion driven instability, and under the presence of the thermal gradient, this where it has a let me just show you one more time quickly. Where is this? Yeah, here. So here you could see that when I chosen it was a spiral wave, then in the presence of a same sign and same magnitude of the Sore coefficient, which means that both species is moving in the same direction, then we realize the drifting of the spiral wave. But at the same time, when I choose species which is moving in opposite direction, meaning the Sore coefficient has the opposite signs, then we realize no drifting of the spirals. Rather, we find out a stable spiral. So somehow it balanced out the this kind of gradients, and it creates a stable spiral waves.
Okay.
Uh,
And what about quasi-periodic patterns? Can they be predicted?
it might be, but since I didn't do any nonlinear analysis, I'm sorry that I won't be able to go through the details of how actually it will affect. But I'm sure it should affect. It should affect in many various ways. Just like here, we could see different variety of patterns coming in quasi-periodic states also can have have some impact.
Okay, thank you.
Okay. Uh, Prashant, now you can raise your question.
Yes, sir. Thank you, ma'am. Thank you, ma'am. Thank you, sir. Ma'am, I have. Is it audible, ma'am, for you?
Yes, yes,
Uh, Ma'am, that the starting of the lecture, you have talked about like breathing spiral. So you
yes.
told that the frequency of those breathing spiral would be equivalent to the frequency of illuminating light. So is it, ma'am? I
Yes,
didn't.
yes. So, if I just briefly, because I didn't discuss this work, but
Okay.
just to show you the reference again, you can go through it. Which is detailed analysis there. So here, what we see. So basically, this is a spiral wave. Let's say, and under a periodic illumination around a where there is a temporal forcing. So this forcing has a frequency. So this spiral breathing like dense to sparse, which basically resonate with the frequency of this externally applied temporal forcing. So the frequency of this breathing is almost equivalent to the frequency of this externally applied forcing through the periodic illumination. As long as there is disparity, it shows a breaking of the spirals, and even the destruction or even complete elimination. So some sort of resonance happens in some can, and the detailed analysis, the analytical scheme by nonlinear multiple time scale analysis is given in this work. If you are interested, you can have a look on there. So you could we can realize all the conditions where this spiral withing will happen, like this amplitude changes, as well as when it will break up. Is it fine,
Uh,
Prashant?
Prashant,
Hello, ma'am. Is it audible now? Yeah,
Yeah,
ma'am.
yeah.
Uh,
So
so I was asking that what would be the length scale of these patterns? What are the forming here?
okay, so
And
these patterns what I have shown, these are of millimeter scales. These are performed in millimeter size reactors, like chemical reactors, which is CSTR. So these are the setups that I showed you briefly in like a slab. So, but you can realize it maybe in a little bit bigger length scales also, like the BZ patterns you could see in the beakers. So those are definitely more than a millimeter.
okay, so mainly they are liquids, right, ma'am? Not
These are
solids.
solutions. These are
These are
solutions.
solutions. Okay,
These are
ma'am.
solutions.
Okay, ma'am. Because there is
Solutions, but
study.
in 2D setup. So we performed the setup is like that, that the system will be in it, performing only the 2D diffusion.
Okay, ma'am. Okay, ma'am. So like
So
because
no
there is a
convection is
study.
considered.
Ha ha ha. Okay, ma'am. Ma'am, there is a study on like creating a special kind of materials, like which can give special type of non-linear optical characteristic. They can show, they used to say negative refractive index material. But I think the experimentally there is a some things are there that they are not able to achieve because those the patterns what they want to achieve in the are in the nanometer scale, and there are some kind of special pattern. But while looking these talks, I was thinking that is there any way that one can proceed in that way to locate that kind of pattern, special patterns in the in the three dimension material, so that they can have such kind of materials actually.
Okay, so you are talking about something in 3D.
Yes.
Definitely, there is evidences of 3D spatial temporal orders, everything, but there are other, dispersal events like drifts and other motions can come and make it complicated. But as talking about materials, definitely materials also undergoes pattern formation. Nano size pattern formation is very common, and you could see in materials in catalytic most of this catalytic reactions, nanomaterials, there are some sort of pattern structures appear. But the thing is that definitely there is a time scale and length scales, and one need to what kind of experiments you are doing, what kind of phenomena you are looking for, that all is important. And if you can actually come up with a mathematical framework or theoretical framework where you can basically with the kinetics and the all these fluxes are there or the perturbations are there, if you can come up with this setup, you can also think about and tune the things to make it in your you can change the length scales of your patterns, and maybe it can help.
Okay, ma'am. Yes, ma'am. So, ma'am, you are utilizing that electric field for the pattern formation. So, is it necessary that the ions only should be there? Is there possibility that if we have nanoparticles endowed there, so
But
that
if they are only
nanoparticles?
charged, if they are only charged, then
Yeah.
only it comes as a electric field effect, so they will move according to the this diffusophoretic motions.
Okay. But instead, if I have nanoparticles, though they also will show the reaction for the applied electric field. So, is there possibility to
It
study?
might happen, but now if they undergo a reaction because of the electric field, that is something different. But if they only show the changes in their motion, that is something different. So
Uh,
one need to think about what is exactly happened, whether the electric field actually creating some reactions or converting some state to another state, or it actually only affecting the flux or the dispersal of the ionic species
okay,
or
ma'am.
the
Okay.
species.
Yes,
Okay.
ma'am. Yes, ma'am. But in the case of applied electric field, the pattern formation can be occur in the three dimension also, right, ma'am? Because if I'm applying the electric field in the overall materials, the whole solution itself, so those patterns are going to form in the overall materials in the 3D. So it's not. I
Yeah,
don't think
so
it's
far whatever I discussed is all in 2D.
okay,
So
ma'am.
definitely if you can come up with a 3D setup, definitely
Okay,
there would be an effect.
okay, ma'am. Okay, ma'am. Thank you so much, ma'am. Thank you, ma'am.
So, are there more questions, students? If you have some questions, you can also write it in the chat box. So, there are no more questions, Dr. Ghosh. Maybe they will write to you. Some people are hesitating.
I'm fine with that, no problem.
Yeah,
Questions are all welcome, they can interact anytime.
yeah.
uh, just
It
email
might be straight
is
to
there.
ask. Students, do you need some clarifications? All right. So there are no more questions. So since there are no more questions, I would like to conclude the session by thanking Dr. Pushpita Ghosh for accepting our talk and for accepting our invitation and giving a very wonderful talk on pattern formation. Thank you.
Thank you very much, Professor Sentivalam. It was it's my pleasure to be, contribute to this lecture series, and I really appreciate this effort. Thank you very much.
I will write to you the other details by email.
Thank you.
Bye.
So shall I just close?
Yes.
Okay.
Yeah.